I'm replacing Marisa Grunewald, which should be here right now, but she's pregnant, and therefore that was probably too much of an effort for her to be here. So I'm going to try to be as good as she would be. So I'm waiting for the presentation. So the CBS barcoding is not my work, right? I'm just the tip of the tip of the tip of the iceberg here. There are many people and many efforts that have been done, and all those people here have been working, but there are many more that have been involved in this. And I'm only going to talk about the yeast part and not about the fungal part. Well, I mean, yeasts are fungi, but you know what I mean. So we have many more data than just that, right? So, in fact, we've been, well, we have about 10,000 strains at the CBS and about 2,000 different species names in terms of yeast. So, if we look at the large subunit, we have a total of 21,000 sequences associated with those strains, those yeast strains. So this is only yeast data again. And CBS sequences only, so not gene bank related things, it's about 15,000 sequences. And the number of strains that have been validated, meaning that the name has been confirmed using the LSU, is about 6,300 strains, while 385 strains have not been confirmed using the 26S sequence. And here are the data for the ITS. So you can see that we've generated about 13,000 sequences during the barcoding project, ITS sequences, and that about 5,000 strains have been validated using ITS. So now I'm going to say a little bit something about the CANDIDA problem, you know. So I'm going to be the evil guy, probably, and you will be able to kill me after if you want. So, and I'm a gorilla, or a chimp, whatever. So here, when we describe new species, you know, we usually have two type strains, of course, and over time, when the strains are coming, the species concept is evolving like this, and so species tend to grow, and further, of course, and depending, of course, on the clades, because, you know, if you are talking about very famous strains or species, of course, we have a lot of isolates, and therefore things tend to grow, and this is the way it's going. So over time, the species concept means something else most of the time, because there are more individuals involved. So when you have things that are close to two groups, where do you assign those two things? Normally, you should look at the type, of course. That's the only one that should be accounted. But the problem, when you use BLAST, for example, using a gene bank and things like that, most of the time people are not taking care of the type. They are just taking the most similar one, and the most similar one might be the one in red here, or, you know, the other distant thing here. So, doesn't work. So, doesn't matter. Anyway, so this one here might be taken as the reference, while it's only a strain among many others. So it should not be the one you should take into account. It should be this one or that one, right? But very few people are really doing those kind of things. So that's a real problem. So when you have strains that are filling the gaps like that, you know, the species are becoming closer and closer to each other, and at the end, you have the lumping solution, which is to just put everything together, you know. And this is what happened here. This is what happened with CANDIDA. That's not the only reason why this happened like that. It was also a garbage genus. But, you know, you see here the evolution of the number of species over time, starting in 1923 up to 2012. In 2013, we had again 30 new species of CANDIDA, and in 14, only one, and in 15, one. And I hope that people have understood that CANDIDA is over and that should be killed, right? Because this is a tree, a very bad tree, I agree. I know that we cannot align those kind of things, so this is based on pairwise sequence alignment. It's not a multiple alignment tree, but still, This is Candida, the clade with all the Candida species and type strains that we have. And you see here, this is the place where the type strain is, right? And the distance, the maximum distance is 100%, meaning you cannot align them at all, right? So using neither ITS, neither 26S. So they have absolutely nothing to do with each other. So it's really time to go for a more splitting solution, right? And I'm going to show you here one tree, which is, to me, quite interesting, although it's yet another big tree like the previous one. It's a tree which is based on mushrooms that have been isolated in Italy at the Venice herbarium. And there were many specialists, amateurs, but still specialists, that have been identifying those beasts on the basis of morphology. And you can see, based on the colors here, so different colors here are different, different species, and the same color, same species. And there, this is the genus level, so when the same color is the same genus, and here is the family. And you can see very well that the clustering is based on ITS, but the coloring is based on the morphology. So the morphology and the ITS are fitting very well together. So even at higher levels, ITS is not completely stupid. So it's just another argument just to say that even if we only have ITS, we have still something quite interesting, right? And if we would have, of course, on top, 26S, ITS, 5.8S, and a few more genes, that's much stronger. Of course, in yeast, we don't have the morphology that they have there, but still, you see the point. So now I'm going to show you a little bit some of the statistics we got from the barcoding project at CBS. And what we did was to isolate the centrality out of the different clades that we had in the collection. So for each species, we defined a central point, either by using the type strain or by using the most central sequence when the type strain was not available, right? So, and we were calculating the most distant strain or the optimal threshold to be able to distinguish all those groups from each other and to cluster them ideally according to the actual classification, to try to fit as best as we can. And this is what we did here. So this is the results for ITS, and this is the results at the species level. So if we want to cluster the strains at the species level using ITS, we'd rather have to have an optimal threshold of 98.91%, meaning that everything which is more distant than 1.09% should be another species for ITS. And that's an average, of course, on all those strains that have been analyzed. And you can see that the best quality we can get is about 80%. So if we have a lower threshold, you can see to the left, we have a lower quality, meaning we are fitting less to the actual classification that was done by the taxonomist. We need to redo this job, of course, after the reassignments that is going on right now. And that will change the picture again, of course. We will be better after that. But still, it's giving you some kind of indications. Here I'm skipping this one. And here we have, again, the same kind of measurements, but for LSU. And here you can see that this is 99.71. So this shows that LSU is less variable than ITS. But there the quality is lower, slightly lower, but still lower. And this is an indication that the 26S is not really able to distinguish all the clades properly. I mean, all the species in some of the clades anyway. So that's the kind of message here. And we've done the same exercise at the genus level. So for ITS, we tried to find the best possible threshold to distinguish the genera. And you can see that ideally the optimal threshold is about 89%. So 89% is the ideal threshold. But here we were including things like Candida, Cryptococcus, and those kind of things that are really, really, really badly defined, that they mean nothing, right? And therefore you can see that the quality of the analysis there is rather low. It will be extremely interesting to do it again when the reassignments will be done in terms of genera, and we will see a huge difference there for sure. But as a guideline, you can see that if you are beyond 11% of distance, you probably are in another genus, right, to a certain thing, to the type of that particular genus. And this, again, on average. In Candida, you could be as far as 100% and still it would still be good, but it's over. And for LSU here, we have those kind of results, and this is 96%, the optimal threshold, and that makes sense again because LSU is less variable than ITS. So these are obvious results, but here it's objectivated by a large number of data. And so we could use this potentially in the future. And here, I don't know if you can read, and the point is not so much to read, but we have those statistics for each of the genera. And we can see where it's more of a problem. And you may be able to see that here, for example, the Baryomyces, Trichosporon, Saccharomyces, and those kind of things are very high in terms of ITS, meaning that ITS is not very variable in those regions. And here we have the same kind of statistics for the LSU, and there it's even worse. And you can see that the very well-known Baryomyces, for example, is not very distinguishable, or using the LSU. And then what we are going to do, we have those statistics that were made by Duong Vu, which is maybe in the room today. And we created a report at the species level and at the genus level for each of the species that we have in the CBS collection. And you can see here, this is for Barnettozyma, this is at the genus level here, this is just an example. What we have is the list of all the strains belonging to that genus with all the sequences attached to them, and then the distance to the centrality to the right, you see, and therefore you can see how good those sequences are attached to that particular genus or to that particular species, depending on which level we are working on. And then at the genus level here, and again we have that at the species level too, we are computing a number of thresholds, which are the minimum similarity, the maximum similarity, the median, the average, and all those kind of things. And this gives you a kind of an indication when you are in this clade of the type of level of similarity that you need to use to decide whether that unknown sequence is belonging to the existing pieces. And here, for example, this is 94.61 instead of 99.71 on a global scale. So that's quite different in this clade than in other clades, meaning maybe we should revise this clade, for example. And here, this is just a tree. So in this report, and all those things will be available via Mycobank. So at the species level and at the genus level, you will have that. And here, you can see here, this is the optimal thresholds, and you will have those trees published automatically and available to you for, for your own information. So one last thing that I'm going to say is that ITS can be extremely interesting for identification, but then we have those kind of double peaks problems because it's a multi-copy locus. And you can see that the copies are really different, and there are many different types of copies within the same individual, right? So this is a very serious problem for next generation sequencing things. So if we want to use ITS for next generation sequencing, we need to know that we are going to overestimate the diversity if we are going to use this ITS. So that's a problem there. That's a real serious problem. And then, but on the other side, for strains typing, that could be a huge opportunity because, you know, the different strains have different sets of copies of the same locus, and therefore we could use it as typing. So instead of having to do multi-locus typing, you know, sequencing many different types of genes, here we would just have to sequence one region, and we would get the profile of that particular strain. That could be very useful in epidemiological studies, for example. So there are pros and cons, but ITS is probably not the only good thing for next generation sequencing. We probably need new loci. And that's it. 15 minutes.